Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on assimilation of food. Now the first question comes in our mind that what is the meaning of assimilation of food? Now to understand this concept, we need to understand the process of digestion and absorption. Now here in the figure, as you can see, I am showing you the lumen of intestine, and this is the, as you can see, this one is the lumen of the small intestine. And when the carbohydrates are completely digested, the digestion of carbohydrate yields glucose, galactose and fructose. Now the question is where this glucose, galactose and fructose are going. That is the question. Where in our body these three are going and how our body is utilizing these three molecules. Then digestion of protein produces amino acid, and then there is a digestion of lipids, dietary fat. So the lipid digestion product, fatty acid, monoglyceride, cholesterol, lecithin, where these all nutrient molecules are going and how our body is utilizing these molecules. All this discussion, all this thing that we are going to discuss under the heading assimilation of food. So first we are discussing the assimilation of carbohydrate, then we will discuss the assimilation of lipid, and finally we will discuss the assimilation of protein, that is the amino acid. So now we are starting the lecture. So here this is the lumen of small intestine, and this cell is the small intestine epithelial cell, enterocyte cell. And here you can see the glucose molecule in the lumen, galactose is there and fructose is there. Now in the brush border, this is the brush border, and in the brush border the transporter molecule is SGLT1. Now we know the full form of the SGLT1 is the sodium glucose co-transporter molecule. So this SGLT1, we know that how along with Na plus ion, due to the electrochemical gradient, this glucose is entering into the cytoplasm of enterocyte cell. Now from the cytoplasm, this glucose is transported outside by another carrier protein, that is GLUT2. And this is the blue color interstitial space. So in the interstitial space there are now glucose. The same procedure is followed for the absorption of galactose. Same carrier proteins are involved, SGLT1 and GLUT2 for galactose also. And for fructose, transporter is GLUT5. Now fructose is in the cytoplasm. From the cytoplasm, it is coming out in the interstitial space with the facilitated diffusion with the help of carrier protein GLUT2. So now in the interstitial space there is a glucose, there is a galactose, and there is a fructose. So all three, these three, where they are going? So all three now they are entering into the blood capillary. So here you see the glucose from the lumen. The glucose is absorbed, and now glucose entering into the capillary. In the capillary there is a blood. So we can say now the glucose is in the blood of the capillary. Now where this glucose is going, all the way we are tracking this molecule where they are going. Okay, now this capillary is there. Now one more figure. See, this one is the hepatic figure for the hepatic portal circulation, and this is the GI tract. We know the digestion process takes place in the stomach, small intestine, and little bit into the large intestine. So absorption process mainly occurs in the small intestine and some absorption in large intestine. So in the wall, these capillaries in the wall of small and large intestine, the blood capillaries are there, and blood capillaries, they join together and they opens into the, or they merge into the venules, or we can say simply the capillaries, they open into the venules. Now the venules join together and they form the vein, and the vein that is formed, that vein is known as the hepatic portal vein. So now we can say what is there in hepatic portal vein. So glucose is there, glucose is there. Then galactose is there, and in the blood fructose is also present. Now this hepatic portal vein, so it is supplying the blood, so it is branching, in fact it is branching into the liver. Then this branches again venule, capillary, then the capillary join, then there is a formation of central vein joins together, and there is a formation of hepatic veins. Hepatic veins opens into the inferior vena cava. So now you can understand, so in the liver the blood is coming due to the hepatic portal vein. So what happened? Most of the glucose, galactose, and fructose remain in the main bloodstream, but the excess glucose, galactose, and fructose, they are received by the liver cell, that are the hepatocyte. So hepatocytes are receiving glucose, galactose, and fructose, and most of the part of the glucose, galactose, and fructose, it remain into the main blood stream. So here you can understand glucose, galactose, and fructose, they are transported with the help of hepatic portal vein to the liver, and from the liver there are hepatic veins. So in the blood of hepatic vein, these three are present, and the extra or the excessive glucose, galactose, fructose, they enter into the cell of the liver. Now this hepatic vein opens into or joins to the inferior vena cava, and the inferior vena cava open into the right atrium of heart. So here you see in this figure hepatic portal vein. This one is the hepatic portal vein. Then these are the branches. Then this branches, venules, capillaries, again they are joining central vein, then the central vein joins, and the hepatic vein. So these are the hepatic vein. and the hepatic vein, they open into the inferior vena cava. So most of the glucose, galactose and fructose remain into the blood of inferior vena cava and then into the main bloodstream, main blood circulation, and extra or the excessive glucose, galactose and fructose enter into the liver cell, that are the hepatocytes. Now in the liver, what happens to this three types of molecule? What happens to glucose, galactose and fructose? So here you see this figure. This is the blood circulation with the red color. Okay. Now you can understand that from the heart, arteries emerge out. Arteries are traveling along all the body parts. From the arteries there are arterioles and then there is a capillaries. So from the capillaries, glucose, galactose and fructose, they come out and they enter into the body cell. Okay, so here this is the blood, okay, blood vessel, and from the blood vessel the glucose, you see the glucose. Now where this glucose is going? So glucose enters into the muscle cell, skeletal muscle, and glucose, many glucose molecules join together and they form glycogen. So in our muscle cell, glucose is stored in the form of glycogen. That is one thing. Now all our body cell, from the blood, all our body cell, all the tissues, they receive glucose, and inside inside our body cell, we know there is a process of cellular respiration. So that is the catabolism of glucose. So in the catabolic process of glucose, the glucose is oxidized and there is a production of ATP and formation of carbon dioxide gas. And this oxidation process, we know cellular respiration, the stages of cellular respiration, that are the glycolysis, Krebs cycle, oxidative phosphorylation and electron transport chain. So glucose is oxidized in our body cell and the ATPs are produced. Now from GI tract, with the hepatoportal vein, the glucose is entering into the liver cell. Now in the liver, what happens to glucose? So you see in the liver, glucose is converted, glucose is stored in the form of glycogen. This is one thing. Glucose is stored in the form of glycogen in the liver. Second thing, glucose is converted into glyceraldehyde 3-phosphate. Now glucose can be converted into fatty acids. Now fatty acids combined with glyceraldehyde 3-phosphate and triglycerides are formed. So we can say from glucose the triglycerides are formed, from glucose fatty acids are formed, from glucose glycogen is formed in the liver. This happens to glucose. Glucose is catabolized, glucose is oxidized, glucose is stored in the body cell, muscle cell as glycogen. Then in the liver, glucose is stored in the form of glycogen, fatty acid formation, triglyceride formation. All this happens to glucose. Okay, now two more. Sugar. Okay, what happens to fructose and what happens to galactose? Now here, if you go to Wikipedia site, there is one article for the metabolism of fructose. You can read that article. So the fructolysis metabolism, fructolysis means metabolism of fructose. Now you see what happens to fructose. Under 1% of ingested fructose is directly converted to plasma triglyceride. Under 1% fructose, it is we are obtaining from our diet, or it is produced by the digestion process of carbohydrate, for example the sucrose. So that fructose can directly convert into triglyceride, but this conversion under 1%, less than 1%. Then 29 to 54% fructose is converted in the liver to glucose. So in liver, we are remembering this, fructose is converted into glucose, 29 to 54%. And about quarter of fructose is converted to lactate. Then 15 to 18% fructose is converted into glycogen. So all these points we are remembering for the assimilation of fructose, how the fructose is assimilated in the body, all this information. In fact, you can read this entire article. I am showing you this. This way. Okay. Now, what happens to galactose in our body? See, galactose. So in our body cell and liver cell, galactokinase enzyme, ATP converted into ADP. So phosphate is added into galactose. So there is a formation of galactose 1 phosphate. Now from galactose 1 phosphate, with the help of this enzyme, glucose 1 phosphate is formed. Now this glucose 1 phosphate transforms into glucose 6 phosphate with the help of this enzyme. Glucose 6 phosphate is converted into glucose with the help of this enzyme. So in short, we can say galactose in our body cell and in liver cell, galactose is converted into glucose. Then one more thing, galactose converted into glucose 1 phosphate, and glucose 1 phosphate can be converted into glycogen. So this is what happens to galactose. This is the assimilation of galactose. Only 2 examples I am discussing with you, considering the depth of your syllabus. So with this 2 things we will remember for the galactose. Now we are moving forward. Now this figure, this process is glycogenesis in the liver and glycogenolysis. So this 2 processes, and one more, this process, gluconeogenesis. So these 3 processes, they are also taking place in the liver, but all these 3 processes I have already discussed with you in the lecture functions of liver. So here I am not discussing these processes with you. But you need to remember that these processes are taking place in the liver. Then here in the liver, this is a glucose molecule and how the branching occur and the glycogen formation take place. How the glucose is stored in the form of glycogen in the liver, and also glucose is stored in the muscle in the form of glycogen. So we have already in the previous lecture glycogenesis and gluconeogenesis, we have already discussed these things. Okay, so that's why I am not repeating those things. Now let's talk about the lipids. What happens to lipids? So here you see, now the lipid digestion and absorption, we are now having the idea what the lipid digestion is and what how the lipids are absorbed. So here this is the lumen, and the lumen fatty acids are there, monoglycerides are there. Beside the fatty acid and monoglyceride, there are cholesterol is there, then lecithin is there. So all this, they are present in the lumen of small intestine, and this digested product of lipid, they are covered by bile salt and micelle formation take place. Okay, so from the micelle, all this lipid digestion product, they enter into the cytoplasm of enterocyte cell, intestinal epithelial cell. So in this, in the cell, there is a cholesterol, there is a monoglyceride, then there is a lecithin, lecithin is there, then there is a FFA, FFA means free fatty acids are there. Now all these are esterified with the free fatty acid, and now in the cell there is a formation of cholesterol ester. Cholesterol ester is formed. Phospholipid, phospholipids are formed. Triglyceride are formed. So this newly synthesized lipid, they are covered by apoprotein, and the particle or the structure that is formed is known as the chylomicron. This chylomicron, then they are entering into the Golgi body for the packaging. Then from the Golgi body, the secretory vesicles are released. The process of exocytosis, the secretory vesicle burst out, and the chylomicron, they enter into the lymphatic vessel, and that lymphatic vessel is known as the lacteal. So chylomicrons enters into the lacteal, and in the lacteal lymph is there. Okay, now from the lacteal, where this chylomicrons are going. Now in this figure, the structure of chylomicron is shown. For example, you see this is the lipoprotein or the chylomicron, and in the figure you see inside cholesterol ester is present, then inside triglycerides are present, then on the surface of chylomicron phospholipid cholesterol is there, then surface protein apoprotein, apoC2, apoE, and this ApoB 100, these are all proteins. So chylomicron. Now we know the chylomicron is in the lacteal. So here in this figure, this is the chylomicron, and this green is the lacteal. Now this lacteal opens into lymphatic vessel, and lymphatic vessel opens into thoracic duct. Thoracic duct open into the venous blood. So the lymph comes into the venous blood, and this is how the chylomicron enters into the blood circulation. So this figure, this entire digestion absorption, this is the concept map for the entire digestion and absorption process. This way. Now you see, you follow the chylomicron where they are going. Now these are the first one is the chylomicron. Now in the structure of chylomicron, 3% phospholipids are present, then 90% triglyceride, cholesterol 5%, protein 2%. This thing we are remembering, and this is just for our information. Okay. Types of lipoprotein. Now here in this figure, you see this one is the GI tract, small intestine. Now with the green color arrow, you see lymph absorbs chylomicron from small intestine, that is in the lacteal. Then lymph drains into bloodstream, that is the venous blood. Then this is the blood capillary is shown. Now from blood, what happens to this chylomicron? See, in the blood, lipoprotein lipase, enzyme lipase removes lipids from chylomicron, and these lipids, they are stored into the adipose tissue, and the cells of adipose tissue, adipocytes. So lipids are stored in the adipocytes of the adipose tissue, and excessive lipid, so liver disposes the chylomicron remnants, and then the lipids are entering into the liver. So this is the entire concept map I am showing you. Okay, this way. Now let's discuss what happens to triglyceride, how the assimilation of triglyceride is taking place. So from the chylomicron, this is the blood vessel is shown. So in the blood, chylomicrons are there, and in the chylomicron we know there are the triglyceride. Now this triglyceride, they are stored in the adipose tissue. Triglycerides are stored in the adipose tissue. Then you see the glucose is entering. We have discussed this thing, and glyceraldehyde 3-phosphate fatty acid, they combine and the triglycerides are formed. And this triglyceride from the liver, they enter into the adipose tissue, and they are stored over here, the triglyceride. Then the blood glucose, this is one route that the glucose is entering into the adipose tissue, inside the adipose tissue, what is happening? Glucose is converted into fatty acid, glucose is converted into glyceraldehyde 3-phosphate, and triglycerides are formed. So in the adipose tissue, conversion of glucose to triglyceride, that is also possible. Okay, so this is how the lipids are mainly. uh, keeping in the mind the depth of your syllabus, you need to remember how the triglycerides are assimilized in our body. Okay, so this is the lipid assimilation. Now the final part of this lecture, that is the assimilation of amino acid. Now we have studied the process of protein digestion, and that we have learned that the proteins, they are converted into either into tripeptide or dipeptide. Tripeptide or dipeptide, they enter into the cytoplasm of enterocyte, crossing the brush border with the help of transporter. The name of this transporter is PEP1. Okay. Tripeptide, dipeptide, the enzyme act on this tripeptidase, dipeptidase. So tripeptide, dipeptide, they are converted into amino acid. This is one thing. Okay, then due to the digestion of protein, amino acids are also produced. Okay, and this is the sodium amino acid co-transporter, and this transporter is transporting Na plus ion along with this amino acid are also rushing in. Due to the electrochemical gradient, amino acid are also entering in with the Na plus ion in the cytoplasm, and from the cytoplasm, this amino acid, this is the carrier protein, and this carrier protein is transporting the amino acid from cytoplasm to interstitial space. So in the interstitial space, now there are amino acid. From interstitial space, where those amino acids are going, so here you see amino acid from the lumen, they are absorbed, amino acids are absorbed, and they are entering into the blood capillary. So again, capillaries will join venule formation. Venule will open into vein. That vein is the hepatic portal vein. So now we know the same route. So now amino acid is there in main blood circulation, main bloodstream, and the excess amino acid also enters into the liver. So what happens to this amino acid in the liver, and what happens to amino acid which is present in our blood? That thing we are discussing now. Okay. Now, okay. So you see, this is the GI tract is shown. Here is the amino acid, and from the blood, this amino acid, they are entering into our muscle cell, and in the muscle cell, from the amino acid, proteins are synthesized. This is how the amino acids are assimilated in the muscle. Then another route for amino acid, that from GI tract, the amino acid, they enter into the liver. Now this amino acid in the liver, with the help of this amino acid, proteins are synthesized. So many amino acids joined together by various kind of bond. The main bond is the peptide bond, and the proteins are formed. Proteins are synthesized in the liver. This is one thing. Then from the amino acid, keto acids are produced, process of deamination that we have discussed previously. Now from the keto acid, the fatty acids are formed. Now this fatty acid, they can combine with glycerol D3 phosphate and the triglyceride are formed. So here you see the amino acid from amino acid, protein formation is done, and amino acid can be converted into triglyceride, and triglyceride are the lipid. Triglyceride is a fat. Now you see this conversion. Now you see this conversion, amino acid to triglyceride in the liver. Okay, and from the liver, this triglyceride, they are entering into the adipose tissue. Why I am highlighting this arrow, that you need to understand that nowadays people are having the craze, especially the youngsters, they are having the craze for the bodybuilding. They want to develop their muscle, and for that they are many a times they are purchasing this protein powder or the amino acid powders, and they think that their muscle are going to develop with this protein powder. And the seller who sells this kind of powders, this amino acid or the protein powder, they are passing the fake information to this youngster that the protein powder cannot be converted into fat. But you see by your own that the amino acid, they are converted into triglyceride in the liver, and from liver this triglyceride is entering into the adipose tissue. So this myth should be busted that the protein powder or the amino acid, they cannot be converted into the fat. So we are also keeping this thing in our mind. Okay, so this is how the amino acid. Now this is a very useful concept map for you, and with the help of this concept map, during your exam time, you can prepare the entire topic of assimilation of food, carbohydrate, protein, lipid very quickly. So here, see GI tract. From GI tract, glucose, galactose, fructose, where these are going, where triglyceride, chylomicrons are going, where amino acid are going. So you see the amino acid entering into liver, deamination process, alpha keto acid and ammonia is released. This ammonia combines with CO2, formation of urea. Okay, then urea, removal of urea in the urine. Then this amino acid, alpha keto acid can be converted into fatty acid, or alpha keto acid can be oxidized, ATP production, energy production. Okay, so the entire topic you can prepare. Glucose, okay, you follow the arrow, glucose, and here you see, okay, glucose is converted into glycogen in the muscle. So I am showing you this concept map. A minute. This is the entire concept map. Okay, and this is also the big picture of nutrient anabolism. Now this is also one good concept map for you for the assimilation of carbohydrate, protein and lipid. Okay, where the glucose is going, what happens to fatty acid, glycerol, amino acid. Okay, and finally I would like to show you the theory points, all the theory points of this lecture. This is the first theory point. Okay, then I'm directly showing you because all the points I have discussed with you, all the theory points I have discussed. Second point, then this is the third point, this is the fourth point, fifth point, sixth point. We have discussed these things, 7 and 8 point, and last point, point number 9, lipogenesis, conversion of glucose or amino acid to fatty acid, and then fatty acid they are used in the formation, in the synthesis of triglyceride. That process is known as the lipogenesis, lipid formation. So with this we have completed the discussion of assimilation of food, carbohydrate, protein and lipid, how they are assimilated in the body. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.